So here's a lovely image of lysosomal function. So you might have an organelle that is damaged, or it's time for the organelle to be destroyed. Say it's older and its own membrane is starting to break down. Well, the lysosome is going to be responsible for digesting waste within the cell in a process called autophagy, which is basically eating yourself. Auto and phagy meaning eating. So the lysosome will take in particles and digest them, and finally the lysosome will be able to go to the cell membrane and excrete the waste out of the cell. So get rid of the molecules and waste products that aren't required within the cell. Okay, so we spoke about lysosomes destroying unwanted material, old organelle parts, foreign matter, and it can break them down with enzymes within the lysosome. So proteases break down proteins, lipases break down lipids that are no longer required. You can actually have lysosomal diseases. So there's certain inherited diseases where the lysosome doesn't perform its function. For example, there's a genetic disease which is more common in a section of the Jewish population. So it's transmitted by the parents. And it's basically a lysosomal disease where the lysosomes are not functional and you build up waste within the cells. And in Tay-Sachs disease, waste builds within cells, which of course causes disease and death of cells, and also of the body itself as well. Okay, so when we're talking about peroxisomes, they are also responsible for waste elimination and breakdown of fatty acids and what are called free radicals, meaning chemicals in the cell that have been oxidized and need to be cleared out of the cell. Okay, so I would see lysosomes and peroxisomes as organelles. They have their own membrane within the cell itself, and they go around and make sure that the cell is clear of waste products, broken down other organelles, and any ongoing debris that needs to be cleared out of the cell to maintain homeostasis. Okay, so the other function of a lysosome is that a cell has a lifespan, right? So it's not like you typically have a cell for the rest of your life. There's constant turnover of cells in the body. So whether we're talking about cells in your digestive tract, your skin, all over your body, the cells are constantly dividing, and old cells are dying. So they have a lifespan. And when it's time for a cell to die, it's digested by its own lysosomes, right? And that process of timed cell death is called apoptosis. So apoptosis isn't a bad thing. It's a way of clearing out older cells and for newer cells to continue on the function. So programmed cell death is apoptosis, and that's typically performed by lysosomes. Okay, so what about this next organelle called the endoplasmic reticulum? Okay, so you can see that the endoplasmic reticulum is just basically layers of membrane, right? Just folded upon itself. And there's the rough endoplasmic reticulum because there's ribosomes on it, those circular structures that are responsible for mRNA translation. And then there's the smooth endoplasmic reticulum, okay? And you can see how close the endoplasmic reticulum is close to the nucleus, right? Because that's where the mRNA is coming out of. It's going to go to the ribosomes on the rough endoplasmic reticulum, sorry, and synthesize proteins that are going to be translated from the mRNA, okay? The smooth endoplasmic reticulum is responsible for lipid production and phospholipid production, and also some steroids are produced in the smooth endoplasmic reticulum. I would think of it as where certain lipids, such as phospholipids for the cell membrane, are produced. Okay, so the Golgi apparatus, again, this is all part of this packaging system in the cell. It's pretty amazing how, you know, the cell can release mRNA that can then attach to the ribosomes on the rough endoplasmic reticulum, produce an assortment of different proteins depending on which mRNA is being copied. The proteins are then packaged into vesicles. The Golgi apparatus takes over and does all this packaging of proteins, sends them to the cell surface, and releases them into the extracellular fluid. Okay, so the Golgi apparatus is the processing, packaging, and shipping. Okay, this kind of reminds me of Amazon, where there's the computer, which would be the nucleus, telling the system what to make. It sends a signal, which would be the mRNA. The signal goes to a factory plant, which would be the endoplasmic reticulum. The signal would be copied, and the proteins would be the shipment, whatever was made. It's packaged in the Golgi apparatus and then shipped to different parts of the body, and often shipped out of the cell into the bloodstream, right? So and it's doing these with these little vacuoles, right? These little packages that are leaving the Golgi apparatus and going to the cell membrane. So pretty amazing. I'm not sure if you got that Amazon analogy or not, but I'll leave it in there. Okay, so the nucleus, which is the control center of the entire cell because it has the genome, right? So the DNA it makes up the genome. You have 23 pairs of chromosomes in your nucleus, so 46 chromosomes. They come as 23 pairs, and they are being copied, right? And when they get copied, they send mRNA out into the cytoplasm. So the nucleus really is the director of cellular activity, right? It's the DNA in the nucleus is responsible for cellular replication and division, so that would be mitosis. The DNA is double-stranded DNA. It's a double helix, it's called. And as we've spoken about before, it's made of nucleotides that pair up with each other, right? So purines and pyrimidines, and we'll go into that in more detail. So the base pair adenine pairs with T, which is thymine, and guanine with cytosine to make the double helix. Okay. So basically, the DNA, which is made up of thousands and thousands of nucleotides, those A, T, G, C, they are what's making the mRNA to go into the cytoplasm, right? And to be copied to make a particular protein. And there's so much information in DNA that there's potential to make almost an endless amount of different proteins. Okay? Remember, the mRNA uses uracil instead of thymine as a base pair. Don't know why. I couldn't explain that to you. I'm not sure if anybody knows why it uses uracil. It just does. It uses adenine, guanine, and cytosine, and uracil. Okay, so what else can we talk about on this slide? So there's 3 types of RNAs, and we've done that. It's the messenger RNA. There's RNA actually in the ribosomes that attach to the mRNA to read it, to copy it. And then there's the transfer RNA, which is attached to the amino acid that's going to become part of the protein that's being produced, right? So I would say the most important one is the mRNA, but all of them have a function in the production of proteins. Okay, so this image we've seen before with the different RNAs, right? Ribonucleic acid. So you have the mRNA, which is from the nucleus being translated by the ribosomes that have their own rRNA. That's not translated though, it's just the mRNA. And then finally you have the tRNA, which is the transfer RNA that brings in the specific amino acid that matches up to the 3 base pairs. Okay, so it really is an amazing system. One amino acid is coming in on a transfer RNA because of 3 base pairs on the mRNA that's coding for that amino acid. And it's gradually making a protein. So a peptide is just another term for a small protein. And as it gets bigger, just call it a protein. Okay? Okay, so this cell is just giving you the difference between transcription and translation. Transcription takes place in the nucleus, where a strand of DNA is copied into a new strand of RNA or mRNA. And translation is the messenger RNA that's being copied by the ribosomes into a protein. That's the process of translation.